thanks everyone for coming early in the morning and fighting your jet lag and stuff. Hopefully you've had your coffee fixed so you can stay awake for the next 40 minutes. So again, as Alex said, I'll go through some of the theory and the applications that we have for like about 20 minutes. We'll run through a code, that a Jupyter notebook that we wrote specifically for this topic. I'll walk through that code for the next 10 minutes or so. You don't have to follow me, but it'd be nice if you can download it as well and kind of follow through using your own computer, download it using Google Colab if you want to. And we'll encourage all these speakers to have some kind of a strength and limitation page, slide, so you kind of understand where everyone is coming from and what one mode is useful and what it is not useful for certain contexts. All right. So I'm going to, again, as Alex said, I'm going to talk you through about what we call as geometric eigenmodes, which are eigenmodes derived from cortical geometry. And I'll explain and unpack all of that in a sec. But I'll start with kind of the theoretical foundation of where these geometric eigenmodes came from. I always like starting with this kind of picture because a lot of us are fMRI people and we've seen these images time and time again, especially those who try and do motion correction, those kind of pre-processing. So you've seen these images for a long time. And I'm sure you've kind of noticed that most of the time this activity propagates smoothly in space and time. So the idea is at these scales of the brain, in the millimeter to centimeter scales, you're pretty much using a camera that can capture these smooth variations. You can't look at the single spiking of neurons. And obviously, this is not fMRI specific. You can see this in different modalities, such as classroom imaging or optical imaging, and also in different species, and also in different types of fMRI, not just resting state. So the idea is there a theory that can predict or explain these phenomena? And I always like to lean towards Peter Robinson's work over the last several decades on what we call as neural field theory. So neural field theory is this compact mathematical description of this continuous propagation of activity in space and time. So the idea is that neural field theory simplifies our concepts of connectivity. What do I mean by that? It emphasizes a lot on what we call as local. Basically, if you look at a laminar structure of the cortex, you will see these horizontal local propagation, local connections, and majority of the connections in the cortex are these local lateralized connections, and also you have those connections that go through white matter that connects one column to the other. But most of the connections are these local and lateral connections. And this concept of EDR. So what is EDR? EDR is what we call as the exponential distance rule. So basically, if you look at diffusion MRI data, and if you just plot the strength of connections as a function of the distance, say the fiber length or simple Euclidean distance between regions in the brain, and that's the x-axis, and then the weight is in the y-axis in logarithmic scale, you will get this like linear curve, and that's signifies an exponential function. That means points in the cortex that are close to each other will tend to have stronger weights as opposed to points in the brain that are far from each other. And it follows this distance dependence rule and specifically an exponential. And this is again not human specific. People have seen this in other species such as marmoset, and it's not diffusion MRI specific. In marmosets they've seen this in track tracing. And again, it's a generalized kind of description of connectivity because you see this in other animals such as mouse, macaques, and other types of mammalian species. So neural field theory really leans on this really simplified concept of local and exponentially decaying rule connectivity. Moreover, it approximates activity as this damped wave that propagates through space and time. All right, I'll unpack this slide a little bit. What do I mean by a damped wave? So if you can see that kind of, I don't know, GIF, I don't know now how to pronounce it. Uh, Imagine like dropping a pebble in a pond or any kind of water material. You will see that you will have kind of wave that propagates from where you've dropped the pebble, but the amplitude of that wave kind of dissipates through space, right? And that's what we mean by a damped wave. And you can also see that in this kind of spring cartoon, you know, if you've done physics in your high school, and if you kind of pull this box that is attached to a spring, it will oscillate, and then eventually it will stop, right? That's what I mean by a damped wave, because the energy kind of just dissipates. Mathematically, you can describe it through some differential equations, if that's your flavor of the day, through this equation shown in here. And that parameter there, gamma, is the damping rate that describes the phenomenon that I've described with the pond. And you also have information of how kind of far these waves propagate. But I'll unpack this equation later on. I just want us to focus on this term. So this is what we call as the Laplace-Beltrami operator. And this kind of encodes all the spatial relationships within that system. And why am I highlighting this? Basically, this is where the geometric eigenmodes kind of arise. So if you ignore all the temporal properties of this equation, basically, you can just look at the spatial profile dictated by this operator. And that's where the geometric eigenmodes come into play. So you can convert that equation, disregarding all the temporal dynamics and so on and so forth, and you will arise with what we call in physics as the Helmholtz equation, shown in here. And again, you have the Laplace-Beltrami operator in here. I will describe in the next slide how we construct it. But that's the one that encodes the cortical geometry. And if you solve this simple, it's like an eigenvalue problem. If you solve this eigenvalue problem, you'll have these sets of eigenvalues, lambda, and you'll also have these sets of what we call as eigenmodes. So basically, you have a pair of eigenmode, eigenvalue, a typical, scenario for eigenvalue problem. And again, if you have like a 1D string, you'll see that these eigenmodes are the patterns that you see. That's the second mode, and that's the third mode. So that's basically the patterns of the modes. All right, how do we construct this Laplace-Beltrami operator? Okay, first, the only thing I need is T1 weighted MRI and some smart tools to construct or reconstruct the geometry. So you can look at it first from the cortex point of view, and geometry will be encoded by the cortical surface. And we have a lot of advanced methodologies, based on free surfer, for example, to segment the brain and extract this cortical surface. You can also look at volume structures, volumetric structures that can't be easily defined by these, simplified surfaces. So say you have the volume of the thalamus. So the idea is you just take a mask of that structure, and that's exactly what I'm doing in here. So the I don't know, gray, or maybe I'm colorblind, whatever that color is, that's just a mass of the subcortical of the thalamus, and I would have this image, which is literally just in voxel space. It's three-dimensional voxel space. So again, in cortical surface space, you have vertices that are connected to reconstruct that surface, but in volume, you only need just the volume. It's exactly the voxel space volume. All right. Again, I promise this is the second to the last slide with mathematics. But on the left is if you're really a hardcore mathematician. This is how you define the Laplace-Beltrami operator. But for a simple human being, as myself, I would like to deal things in approximations. Obviously, cortical surfaces are not continuous entities, right? They're inherently discrete. You can only sample the space up to a certain limit. So you have what we call as mesh approximations to the Laplace-Beltrami operator. Basically, what it's doing is just looking at kind of the nearest neighbor, the neighbors of the points and calculates some cotangent angles. And again, that is a high level of description. I'd encourage you to kind of go through the codes later on to really look at how these are calculated. And basically, that will approximate this operator, basically the operator that describes the change in space along this cortical mesh that we have. All right, suppose we've now absorbed all of this and we really now know how to construct the LBO. What do we actually get? So in the cortical surface, you will get these sets of eigenmodes. And they are arranged in a unique manner, where the first couple will be corresponding to what we call as low spatial frequency or long spatial wavelength modes. Basically, what it means is that the spatial pattern kind of covers the entire, all the modes cover the entire brain, but the variation is on a larger scale. As you can see here, for example, it just changes from the rostrocaudal axis. But as you move further along this mode, the family of modes, you'll get this high spatial frequency. And again, this is a natural ordering of the modes when you calculate it directly from the Helmholtz equation. So there's no, I didn't reorder it accordingly. It just inherently is ordered in this way. And again, you can get the same thing in voxel volume space, for example, here in the thalamus. And yeah, you can see that every single point there is a voxel. So depending on what space your favorite is. All right, so what is so special about these geometric eigenmodes? One thing is that these eigenmodes form orthogonal basis sets. What do I mean by that? These modes are independent with each other. That means you can't represent one mode using other modes. However, you can, so now using this concept, because they are orthogonal and linearly independent, you can now represent any map defined on that surface that you have. Okay, suppose you have this map Y. You can now reconstruct that map in terms of a linear combination of all the modes weighted by some coefficient A. Okay, that's for a spatial map. You can also do this in maps that also vary in time. So basically, what you'll do is for every single time snapshot that you have, you do this decomposition. And eventually, you'll get this, say, for a spatial map, you'll have I'll call it a spectrum of the coefficients. But for a spatial temporal map, you'll have the spectrum of the coefficients for every single time point. Okay. Let's unpack that linear decomposition again further. And because this concept of orthogonal linearly independent basis sets is not completely new. If you're familiar with Fourier transform, it's the same kind of idea that in Fourier transform, you unpack a signal in terms of functions that are linearly independent. In the Fourier expansion case, the functions are sinusoids that have specific frequencies. So it's kind of the same analogy. If you really want some further analogy, it's the same concept as colors, right? You have a specific color, but the linearly independent basis set there are the RGB or the CMYK. And by combining the RGB and the CMYK in various combinations weighted differently, you will arise to a different color. So it's the same kind of concept. All right, let's see if these kinds of decompositions actually work in the brain. Because if not, then what's the point of me being here? All right, let's start with kind of fMRI decompositions. So say here is a group average map from the HCP data set for every single task that you have. Are we able to use a small subset of the eigenmodes to reconstruct this map in an accurate manner? And pretty much, yes. So you can use about 100, and you can almost see that the maps themselves are almost perfectly reconstructed. And you can do this methodically and increase the number of modes, for example, y-axis is the accuracy, which is literally just the correlation of the reconstruction and the data that you have. And on the y-axis is the number of modes. And if I keep on increasing the number of modes, you'll get, the accuracy keeps on increasing and increasing until you get to some level. And again, this is just a subset of the modes, about 100 and 200. And yeah, and in our previous work, we've shown that if you kind of compare these types of reconstructions with other types of basis sets, it seems to be performing quite well. All right, because this is an educational session, I will startle you with some learning polls and see if you really grasp some of the theoretical foundation that I've kind of mentioned over the last 10 minutes or so. All right, the first question is, what do you think is the order of the geometric eigenmodes reflecting? Is it the amount of variance explained? Or is it just independently ordered according to some spatial frequency? Who says A? Who says B? I think I've succeeded, so definitely it is B. So again, the modes are ordered naturally without any fiddling around, and it only represents the spatial frequency from low frequency to high frequency. I have another question, and it's something that I didn't mention. How many modes do you think are there, say, for a cortical surface, say with, 100,000 points? Is it A, equal to the number of vertices that you have? Or I can have as many as I want until I die. Who says A? And who says B? Good point. So it looks like I didn't succeed. So the number of modes that you have is actually finite, and it's only, the maximum is the dimension of your system. It's literally the same as doing a PCA, right? If you have 3 dimensions, you can't have 100 PCAs, and you can't have 100 PCs. So it's the same concept in here. It's equal, the most number of modes that you can get is equal to the dimension of your space. So in a cortical surface, it's the number of vertices that you have. All right. Okay. All right. Let's move to the next bit. How do you now use these modes to explain various concepts of brain structure and function? So I'm going to show you some of the applications that we have used. The first thing is, can we use these geometric eigenmodes to represent what we call as functional gradients, which Boris will explain later on how to calculate. So basically what we found on the left is if we just calculate the modes of the geometrical structure of the thalamus, just the in-volume space, and we calculate the gradient using its functional, intrinsic functional connectivity, we see that these modes and gradients align perfectly, almost perfectly well. And the same concept is true for other structures that we found, suggesting really that potentially these geometric eigenmodes really the geometry of these volumetric structures play an important role in shaping the functional connectivity within these structures. The next thing is dynamics. All right, I've shown you ways to reconstruct data in various ways, but geometric eigenmodes are not just for data reconstructions. You can also make predictions of dynamics. Why am I saying that? Is because I will go back to how I started, because again, the geometric eigenmodes are inherently embedded within the neural field theory concept. So I'll now go back to this equation. So again, this equation describes the propagation of activity phi in a location R in the cortex at time t. And again, this equation shows the evolution of that activity when you have some certain input Q. And Q could be anything. It could be a structured stimulus. It could be activity from other populations in your brain, say the thalamus or any other subcortical structures. But here we're just formulating it as one kind of input that encapsulates everything, intrinsically and extrinsically. And if we use this model, you only have 2 free parameters. the gamma, the damping rate that you have, and the length scale. And you can match this to some physiological parameters. So what I'm going to show in the next slide is, okay, suppose we have this really simple model of wave dynamics in the brain. Can it actually match what we see, say, in fMRI? And here, to make the problem much, much simpler, we'll fix one of the parameter, gamma, and then we'll just have one free parameter. Because we want a model that is as simple as possible without tuning it into some optimal kind of representation. And what we found is that, say, that's your resting state activity. We found that comparing to other models with more complexity and has more information, and again, the wave model only requires cortical surface and cortical surface. Basically, that's it, right? And that wave equation, we can find that you can match the properties of functional connectivity quite well. You can do that in various ways. Say, if you look at how do I reconstruct every single edge of that functional connectivity, it performs almost equally than other competing models. You can look at it from a node FC level, so you just take the average of the FC, and we can find that the simple wave model performs quite well. And you can also look at further kind of complex measures such as the functional connectivity dynamics, or FCD, and you can see that it also performs quite well. You can also model not just resting state dynamics. You can also model some kind of simplified, say, visual evoked activities that is being shown here. All right. Okay. I have 5 minutes. In the previous slides that I've shown, it's treating the cortex as this homogeneous quantity, right? That every single point is the same in the family. It doesn't really change much. Every single point has the same behavior. That means the wave can just propagate kind of isotropically. But we know that in the brain, it's not always the case. You have variations in the brain based on some architecture, myelination, gene expression, receptors, etc. So I'm showing here the case where you treat the cortex as this homogeneous quantity. And we have a really smart student in the lab, Victor, who's thought about how do these heterogeneities, such as myelination, could affect the structure of these modes, and how does it change the prediction of brain dynamics. And basically, you can do that by modifying this equation and incorporating this term. It's like a diffusion tensor that dictates how the spatial curvature will be affected by those heterogeneity. And what you can do is, say, get a T1, T2 map that you have and incorporate that to the definition of the diffusion tensor. I don't have the time to explain that late in this talk, but we can chat later on how do we construct that diffusion tensor. And you can see that the modes will change, but for those who are really awake and have had coffee, you can clearly see that the first couple of modes doesn't really change much. Basically, the idea here is that heterogeneity doesn't really change those long wavelength, low frequency modes. The reflection of this heterogeneity will be seen in those much higher frequency. How about dynamics? Basically, what we found is that if you use these heterogeneous geometric modes, it can predict dynamics much, much better. And here on the Y is just a combined metric that we have. But basically, the increase is about, 30%, 40%. And that is true for like most of the maps that we have, say T1, T2, and layer-specific genetic profiles. But some maps doesn't really affect the dynamics much, much higher than the others. And what Victor has shown that this is not just the case for humans, it exists also in other species that we have, macaques and marmosets. If you're interested on this thing, he has a poster on Friday and Saturday. Make sure you visit that poster. Right, so that's dynamics, and effects of heterogeneity. How about structure? We also have another, I mean, all the students in the lab are smart. We have a smart student in the lab, Francis, who's developed this mode-based model to predict the connectome. Again, I don't have the time to explain how this equation has been formulated, but basically his idea was the connectivity between locations I and j will be defined by some coupling between the modes psi m in those locations, but scaled appropriately by some length scale r, which is the same as the equation that I showed a while ago, and the eigenvalue, basically the spatial frequency. So basically the idea is that at certain frequencies, these modes will couple with each other, and it kind of enhances resonances of the dynamics between those two, which suggests that maybe this is where the connections would exist. And what he's shown is that again, this is probably one of the first vertex level models of the connectome. He can show that it can predict a lot of the topography and the topology of the connectome in both vertex level and parcel level in humans, but also in other species. And again, if you're interested in this topic, I'd encourage you to visit that poster and his talk on Saturday, which is unfortunate. It's after club night, but make sure you visit that talk. And finally, another application that we have is how do we kind of define boundaries in the brain using geometric eigenmodes. So our simple idea is that to use these modes and its nodal lines to define the boundaries. For example, here we do an iterative model where you cut the brain according to the eigenmode, the first eigenmode. And you can, by doing so, you can recreate parcellations of different scales here. And then we compare that to other types of parcellations that we have, say, desiccant, the Glasser or the Schaefer. And basically, by using this simplified methodology, which is eigenmode-based to parcellation, we can find that the parcellations that we're getting are much more homogeneous than all the other parcellations in capturing the properties that I am showing at the top. Basically, it's able to capture them much, much better, except for that thing that must not be named, but no. The Schaefer parcellation seems to be performing quite well. And we've shown that this methodology is generalizable to other structures and to other mammalian species as well. And again, we can discuss later on how we develop this method, but I encourage you to go to Priscilla Levy's, another student in the lab, poster, who can talk you through how we develop these geometric-based parcellations, or you can go to our preprint down the bottom. It's not really informative, unfortunately, but yeah, we have a preprint that you can kind of go through further. I think that's basically the talk, and I'm gonna walk through a little bit of the code. How do I do this? I'll just go through this slide. We have a Jupyter notebook in there, but I'll probably, in the interest of time, I'll just go through what is actually inside that Jupyter notebook. So once you download that Jupyter notebook, there are functions there that are predefined, there are modules that you need to download, and there are example data sets that you can download as well. Basically, the first component of the code is how do we calculate these geometric eigenmodes. It's relatively simple because it's wrapped up in this function that we have. But basically, the only input that you would need is some kind of surface. It could be defined in different space. The example that we have in the notebook is just for an FS average 5 in the 10K, just to make the calculation much, much faster. It could be a definition of the mid-thickness or the white surface or the PL surface. You can choose which hemisphere you have, the number of modes. And I want to highlight this thing. The circle is in the wrong spot. But I want to highlight this thing. So basically, you can calculate the modes in two ways. One is if you want to mask the medial wall here, or if you don't want to mask it. I personally advise to mask it, is because if you don't, you'll lose some of the energy of the modes, which are not informative, because we don't have signal in the medial wall anyway. So you want to constrain the energy just within the cortex that has functional data. And then you can do an eigen decomposition, which I've shown a while ago, using, again, this function that is, I don't want a breakpoint, but yeah, this function that wraps up how you can calculate that eigen, you can do the kind of decomposition of a map that you have and all the modes that you need. It could be the first 100, first 200, it could be everything. Just take note, if you put in all the 10,000, you might need to wait for a while, all right? So just caution. And then you can plot, say, look at the coefficients as a function of the number of mode, and you can play around with that kind of thing. And now, how do we do the reconstructions? So basically what we do is you use these coefficients, and you multiply it again with the eigenmodes, and then you sum them together. And you will get some kind of reconstruction. So you have, say, this is the original map that I have. You can combine the first 5, and you can see that, it's kind of capturing it, but if you keep increasing it using some linear combination, you'll get that the accuracy increases, as I mentioned a while ago. And also, this is not only applicable to spatial maps, you can do the same with kind of FC. So what you'll do again is you reconstruct every single time point, multiply the coefficients to the modes at time point 1. So you now have a reconstruction of the activity at time point 1, and then you do that through all the time points, and you can now do your typical FC calculation after that. So that's what I'm showing here. So this is the original FC of some resting state data that is incorporated in the notebook. And you can see how we do the reconstructions of the FC as in increasing number of modes. The code also have a way to do the modeling. All right. All the codes in our GitHub page is actually in MATLAB, but a little bit of Python. But we've converted all those MATLAB codes to Python, if that is your flavor. So again, in the Jupyter Notebook, you can play around with certain parameters. Say, if you want to change the speed of the propagation into some level, you can do so. You can change that parameter in here. And then you can get some activity of the modes themselves, or this is the combined activity that you have, spatial temporally. And by doing so, you will get this, every single row in here represents the different types of wave speeds that I have. Something typically average for human resting state data. If you want something really, really slow or something really fast, this is the type of activity that you'll get that is produced by the model. Okay, those are the codes. And again, I'm happy to walk you through later on during the break if you need help further. But again, I'd encourage you to go to our parent GitHub page to go walk you through more sophisticated analysis and stuff. All right. Finally, I would like to go to the strengths and limitations. All right. The thing first is the geometric eigenmodes, as I mentioned, are anatomically grounded. You only need T1 weighted MRI, and it's completely independent of the data. That means you can use it to represent any types of data, not just resting state, for example. Similar to the next couple of talks, it's a compact and low-dimensional representation of, in our case, for example, is fMRI data. But I think the more important thing is it's fundamentally linked to some generative mechanism, which is the neurofield theory that I alluded at the very, very beginning. And that's important to highlight. It's generalizable to multiple individuals, species, structures that you have, and has a wide array of applications. But again, I love using this term. All models are wrong, but some are useful. And we acknowledge that the geometric eigenmodes have some limitations, and maybe some of you have already encountered these limitations if you've played around with it. The main problem at the moment is comparing the spatial patterns. And I think a lot of the low-dimensional representations of the brain would have the same problem. It's not, so the idea is if you, say, calculate the modes of one subject, and you calculate the modes of another subject, the first 10, maybe, would kind of match up, right? But once you move to the higher frequency, they would kind of switch with each other, and then you now lose, how do I now compare mode 55 of one subject to mode 58 of another subject if they're actually not the same spatially? And that's inherently a problem that I don't have the answer, and I'm sure smarter people in the room maybe have some answer. You can do some alignments, you know, but part of me feels like if you do those alignments, you lose some individual level information. So I'd caution you on that. We don't have at the moment a framework to combine these cortical and subcortical modes because they're inherently not captured by the theory, but we are trying to work on how we can do those coupling much better. It is obviously, eigendecompositions are inherently linear, so it's difficult to predict dynamics that are nonlinear in nature. So I caution you on that as well. And yeah, and lastly, it's agnostic again to the connectivity that doesn't conform to this local and EDR. So it doesn't capture those long-range connections that you probably have been thinking already. And yeah, thanks to all these people. And just wanted to plug for next year, there's a NUSA workshop happening for brain function, organization, and behavior. If you really like Australia, you can come back next year. Thanks.
Thanks very much, James, for a wonderful overview. I'll open up to the floor for any questions. While you're getting ready to come up to the microphone, I'll just read some off the poll. So one of the questions is there any difference in the results if you use surface versus volumetric input?
I mean, it's tricky. No, I don't know. I haven't checked that. But the cortex is relatively thin, right? So it's tricky to do. You can do the volumetric eigenmodes there if you want to, but I haven't checked. Part of me feels like, no, it won't change much of the modes that you will see.
My question that I had was regarding about the functional connectivity portion of your talk, and I was wondering about the effect of physiological noise and consequently the way that you pre-process the data on the modes that you're getting. Do you have any like comments about the sensitivity of the results and model to the physiological noise?
The model itself doesn't take into account external factors such as physiological noise. It's assuming you have this really pure fMRI signal. So the problem will lie when you now compare the results, the prediction. That's where the problem is, not the theoretical component of it.
That's true, but in practice we know that the functional MRI, the bold signal is dominated by physiological noise. I was wondering how sensitive is the model parameter estimation to those physiological noise that exists in the data.
Again, it won't capture those things, but you can modify the equations a little bit to model the physiological noise and put it not in the parameter space, but more of like you change the input a little bit and you incorporate like a structured noise that is similar to a physiological noise to see if you kind of match your model to your data, if that makes sense.
Okay,
yeah.
Thank you.
Hi, great talk. I have a question around the matching you said about different modes will have different ordering, and sometimes also if you do two cohorts, as you know, that's a heat map starts from negative to positive, goes through. That also switches. Sometimes some networks, like if I'm comparing it to a very specific functional case, functional connectivity case, the network switch, like somewhere the higher order networks will become negative, where the primary networks becomes positive. And the same, another cohort of same age group, the, the data remains, the distinct remains same, but the negative positive just, like what we say, changes. So do you know why or what that mean even?
I think I'd like to highlight that the sign of the modes don't really mean anything. It's literally just saying where the pole of the axis is. It's the same for any kind of low-dimensional representation. The signs themselves don't really, it's not really meaningful. It tells you where the opposites are,
Yeah.
but doesn't tell you that the positive has some magically more informative
Okay.
thing than the negative.
Okay, thank you. Yeah.
All right, we've probably got time for one more question. I'll read off the. This is a spicy one. So, with all the gradient methods providing a coordinate system to look at brain regions, should we still use parcellation? You got 10 seconds.
Yeah. Whew. I'll make this very diplomatic. Yes, I mean, parcellations are useful to some effect. Every single tool that we have is useful for a particular context of interest. So parcellations are useful if you want to really look at localized processes. But if you really want to look at whole brain function and activity, then parcellations are probably not useful in that case. But it is useful for certain contexts, and you just need to make sure that the context that you have requires the use of parcellation.
Great. Thanks very much, James.